Hi everyone, and welcome to the first lab for microbiology. In this lab you're going to be learning how to use aseptic technique for culturing and how to streak for isolation. So the first thing you want to do is find a clear, clean space to do your mock lab, and you should clean it off. Always clean your lab space. So like some Clorox wipes or Clorox spray. These are actually kind of dry, but wipe down your space real good, make sure it's clean. All right, and then also get out your PPE, your gloves, and go ahead and put those on. All right, if you want to get a lab coat of some kind, really look the part, that's fine. You also need to make sure that your dress, your attire is appropriate as well. If you have long hair, your hair needs to be tied back. If you have, I have some little strings dangling here from my shirt, but I tucked them in because I don't want them dangling into my culture. So you just want to make sure that none of your clothing or jewelry or hair is able to accidentally contaminate your workspace. All right, other things from your kit you're going to need. You're going to need your culture tube and lid, and you're going to need some fake culture. So I suggest using mustard or yellow paint with a pinch of glitter in it, and you'll see why. There's a reason for that. And you're going to need a rack to hold your test tube, so you'll have to improvise that. I didn't include anything for that. You're also going to need a Petri dish. So mine has agar in it, and yours does not. But if you want to get some Jell-O and put some agar in it to make it more realistic, that's fine too. You will need your China marker, and you will need your inoculation loop, and you will also need your mock Bunsen burner and your mock biohazard waste. So, aseptic technique. All right, so let me get through here. What we're going to do, what we're pretending to do in this lab, is to inoculate a culture from another culture. So we have a sample of mixed bacteria and a sterile agar plate. So we're going to transfer bacteria from the culture to the plate sterilely, using sterile technique. So we will be inoculating this culture. Then we would incubate it overnight and look for growth. And we're going to use the streak for isolation technique. So our hope is that we would isolate the two different types of bacteria in this mixture. And then in the next lab, we would be doing inspection, where we look at the bacteria under the microscope and also do some gram staining in order to identify them. So let's start with some aseptic technique skills. First of all, handling a culture tube. So this culture is actually sterile. This is just sterile broth in here. It would be cloudy if there was actual bacteria in here, right? But we always assume that our cultures are, have growth in them. So we don't want to get them on ourselves, and we certainly don't want to get our bacteria into the culture. So we're handling it to protect both us and the culture. So when it comes to manipulating a culture tube, all right, you're going to need to remove the cap, and you're going to need to be able to stick the culture loop into the tube. So you need two hands, one for the loop and one for the tube. So you can't just remove the cap and hold it like this. You don't have enough hands for your loop. And you can't put the cap down because it could become contaminated. And it also results in the tube being open for too long. So there's a couple of ways to do this. For people who've been doing it for a while, they often do it one-handed, where they unscrew the lid with their hand and hold the tube open. So I have the lid in my thumb and forefinger, and now I have my other hand to do with what I please. Another way to do it is to remove the lid using your pinky finger of your loop hand. So I have my loop in hand, and now I just use my pinky finger, and I hold the lid and I unscrew the tube, and now the lid is in my hand. I can get a sample, and I can put the lid back on the tube. All right, this is just like a muscle memory skill that you have to get good at. Sometimes in lab I have students work in pairs and one of them pulls the cap off for the other person so they don't have to worry about. But for you guys, you can practice this safely at home doing it yourself. All right, so that's sterilely handling a culture tube. Leave the lid off for as short amount of time as possible and don't put it down. Now for handling an agar plate. All right, we always want to keep the lid on it. So if we open this up, we can see the agar is in the bottom part of the plate, and the lid here is the top part that comes off. When we handle our agar plates, we usually do them upside down. So we put them lid down. Get this out of the way for a moment. Okay, and we never leave the plate open on the bench top. So this is a big no-no. Okay. If I want to open the plate to swab it, I have my loop ready, I'm going to have the plate upside down on the bench top, and I'm just going to grab the bottom with my hand, and I'm going to hold it open, and then I can spread my bacteria on it. I always hold it vertically or maybe a little bit at an angle. I try not to hold it horizontally like this because then I'm just asking for particulates from the air to fall on it and contaminate my culture. So I always try to hold it at an angle to protect it from contamination. I also generally, as a rule, I try not to talk or even sometimes breathe, like exhale, while I have my plate open, just to make sure I'm not getting any of my bacteria on it. Obviously, I'm not being that careful right now for this demo video because I have to talk to explain. And then we incubate plates upside down as well, and that's because in the incubator, there's a tendency for condensation to build up on the lid. And if it builds up on the lid when it's this way, then the water can drop down onto the culture and make a mess of it. So if we do it this way, the condensation that lands on the lid stays on the bottom there. So that's just a fun tip. Bunsen burner safety. Your Bunsen burner is for pretend, but in real life we would make sure that we have nothing flammable around it. Everything's tied back, hair and clothing. And I preset this up so I have the valve opened just to the right place, but usually you start with it closed, and you keep the gas off until you're ready to light it. So I'm going to turn the gas on, hear the gas, and I use the flint to strike it. So you can see the top of the yellow flame here. I usually want my flame to be completely blue. I don't know if you can see in this video, something like white to hold up maybe behind it. I don't know. Can you see the blue? I can't. Anyway, this part right here is all blue flame, and then there's some orange flame at the top here. The blue flame is the hottest, and that's where I want to flame my loop. So I'm just going to hold it in the flame until the loop gets kind of glowy. You can kind of see it glowing there, and I'm going to flame it all the way down to the end of this wire. And I'm wanting it to get nice and orange and hot. So you'll do this. I want to see you flaming yours an appropriate amount of time, even though it won't get hot. All right, now this is a metal loop. This is very hot. If I go straight into my culture, I'm going to fry those bacteria. So I do need to wait and let it cool. I do not want to wave it around in the air like a wand. All right, that would contaminate it. I also don't want to blow on it, so we just have to be patient. Patiently let it cool. If it's too hot, you'll hear a sizzle when you go in there. It's honestly not too big of a deal with a liquid culture, because you might fry some bacteria when you first go in. I didn't get a sizzle, so I'm good. So I'm just dipping the loop right in here into the liquid, and now I'm going to pull it out. Okay, and now we're going to streak for isolation. I'm going to hold this. No, we're not. That was just a demonstration of how to get culture on your loop. And now I'm going to flame it off and continue to go through the protocol here. All right, so just some tips for you when you're setting up for lab, any lab with me. We're going to turn down the oxygen a little. I don't want so much orange in the flame. All right, Always assume gloves are contaminated. Don't touch your face. Don't touch things around your room. All right. Keep your gloves away from your face. When we are writing and labeling our plates, we're always going to label the bottom side, the agar side of the plate with our marker because lids come off, right? And so if your lid comes off and you mix up the lids, then you don't actually know what's on your plates. So you always are going to write on the agar side. We always keep our plates and tubes open for the bare minimum amount of time, like I showed you. We always flame our loop before and after going into culture. And if you are using agar, you just want to be careful, be aware of the fact. If you want, you could actually use Jello. Use like a little cup of, instead of using a Petri dish, you could use like a Jello snack cup if you want to get the feel of what agar feels like, because it's very much this consistency of Jello. So a lot of beginners actually end up scraping it too hard, but that's quick and easy to learn when you're in a real lab environment. All of your waste should be disposed of in a biohazard waste, in your case a biohazard waste, and we always keep our plates inverted. Okay, so a little bit about growth of bacteria in these different types of media. In a broth, if this broth were actually containing bacteria, it would be cloudy looking. There's actually different, we call that turbid or turbidity. So if it's clear, that's a pretty good sign that there's no growth. Some types of bacteria will grow in clumps, so we call that flocculence. So it's not cloudy throughout, but there's like little globules floating around. Some like to grow in a pellicle that forms a layer on the top, like a film on the top, and some will like sort of hang out on the bottom, sink to the bottom. So there's some different patterns of growth. When I went to the fridge to get a clean, sterile tube, I was checking to make sure that none of the ones that were supposed to be clean had any signs of growth in them. So if I have sterile media, I want to make sure that it looks sterile. So the way that bacteria grows in culture is different than how in liquid is different than how it grows on solids. So on solids, it forms colonies. And we can see lots of different types of colonies in this image here. Some of these I think are fungal colonies that look kind of fuzzy. All right, but lots of different sizes, shapes, colors. All right, so there's a little bit more information we can get about mixtures of bacteria when we grow them on a plate. And so what we're doing today is using this solid growth media to get isolated colonies from a mixed culture of bacteria. Two different types of bacteria, yellow and red is what we're going to go with. So remember, a colony is just a spot where one bacteria landed and then grew, proliferated in that spot. So it's really a bunch of clones of the same bacteria. So when we do this streak plate, what we're actually doing is we're taking this mixed culture and we're going to spread it out in one area of the plate. Then we're going to flame our loop and we're going to spread a little bit of that out into another part of the plate, and so on and so forth. So we're going to do 4 of these dilution spreads. And our hope is that we'll end up with a plate where initially the culture grows very thickly and we can't really distinguish between red and yellow, but by the end, it's someplace on the plate anyway, we have some individual colonies of each type. Okay, so when we do that, it's possible then to start pure cultures. We can touch our loop to one of the, in this picture, the blue bacteria, and inoculate a liquid broth and get a pure culture of just that type of bacteria, and then get a pure culture of just the other type of bacteria. So this is a really important technique, especially when you're working with like researching bacteria, and you want to make sure your cultures are pure. So this is the streak plate isolation method that we are using, and I will demonstrate it for you. There's sort of, there's a little bit of creative license here. The old school way is to do it in kind of a zigzaggy pattern, and the new school way is to do it in sort of stripes. So there's 2 patterns here, and I'll see if I can show you both of them when I do it. All right, so the materials you're going to need, I showed you at the beginning of the lab, but your culture tube, I kept this lab the same as if we were doing this in real lab, because I want you to see what we would actually be doing. Okay, so I said that you should have a mixed culture of Coxuria rhizophylla and Serratia marcescens. I color code these because Coxuria grows yellow colonies and Serratia grows red colonies when it's grown at room temperature, 25 degrees Celsius. So that's what we're pretending we have in our culture tube. All right, and you have an agar plate. Okay, so the first thing you need to do is label the plate on the bottom with your initials and the date. Because if for some reason you can't get back to your plate, we need to know what it is and when it was started. So, and you want to label it around the periphery, around the edge. So remember to label it on the bottom, on the agar side of the plate, and do it sort of around the edge. You don't want to like take up the whole space with your labeling. All right. Then you are going to use your pretend Bunsen burner, and you're going to flame sterilize your loop. Okay. So we're going to wait for it to get nice and golden orange and glowing. I don't know, let's say do it for 10 seconds. Count to 10 Mississippi. Okay. And then we need to wait a couple of seconds for it to cool. You can even say, now it's cooling. You know, narrate your actions. Then you're going to want to remove the lid either with one hand or with the pinky of your loop hand, which is usually easier for students. You carefully go in without touching anything else around. Dangle it around in the liquid and close the tube and return it to the rack. All right, you should be able to see a little bubble of liquid in your loop, kind of like a bubble wand. And then we're going to spread that liquid on just like one side of the plate. One up here on the top. Okay, so this top part here. And remember where you did this. If you are using mustard or something, it'll leave a trail. You should be able to see it. So when you do it, get like a good blob of that mustard with glitter in it. All right, now we're going to flame sterilize. What you're going to need to do is you're going to need to maybe get a little paper towel or a tissue and wipe off your loop, because that's what this step does. It cleans off the loop so it's totally has nothing on it anymore. Okay, so you're going to need to clean off yours. Then you're going to turn your plate. Move this out of my way. So now I'm just working with the plate. We're not going back in the tube. We're not getting any more culture out. We're going to open the plate. And before, let's see, I had swabbed it up here, up top. So now I'm just going to go into that section and drag it into another area and just kind of color in this other area over here, about a third of the plate maybe. I'm going to close it back up. So what I've done is I've just taken some of those bacteria in that the area where I actually spread out a lot of bacteria, and now I'm spreading them out a little bit thinner. I'm going to flame my loop, let it cool, and I'm going to open my plate, find where I streaked before, and I see it, and now I'm going to just touch the loop in the area where I had streaked before, and I'm going to spread it out. I'm going to do the zigzag pattern in an area that is clean and has not been touched yet. So I should have this, I don't know if you can see it, you can kind of see where my streaks are a little bit. So the pattern looks something like this. I colored it in one area, and then I flamed my loop, and then I just dragged my loop through that area, spread it out some more, and then you can do it a fourth time as well. So 3 or 4 times is usually sufficient. And hopefully you will see in your plates that your mustard and your glitter, that maybe you got that glitter spread out into little individual dots of glitter. I don't know, I was hoping for a helpful visual. But just note that when you're doing this in a lab setting with actual bacteria, you can't see a thing. there's nothing on this plate. It'll show up tomorrow, and I'll show you some images of some plates that students previously have grown when they did their streak plates, and you'll be able to analyze your results. So when you are done with your plate, you will leave it here. We'll pretend this is an incubator, and you'll say, I'm going to incubate this plate at room temperature overnight. You will flame sterilize your loop and put it away, and you will turn off your flame. Okay, you can take it out of the cameras, I don't care. But do some kind of motion to indicate you're turning off your flame. And actually you should put the plate away somewhere. And then the final thing you should do is clean off your lab station. So get those Clorox wipes back and wipe down your lab bench. And now your lab is complete, and you are just awaiting your results.